right, so yesterday we wrapped up by just a quick overview of the 3 steps in cell respiration. So the first step we're going to talk about today is glycolysis. Glycolysis occurs in the cytoplasm of the cell. The second step is called the Krebs Cycle. We're going to talk about that today also. It occurs in the matrix of the mitochondria, and this is actually going to come up on another slide, but I'll just do a real quick reminder. The matrix of the mitochondria is this center part right here, in here. So that's where the Krebs cycle or citric acid cycle is going to happen. And then the final step of cell respiration is the electron transport chain, and it happens on the inner membrane of the mitochondria, which is also called the cristae. And that is this, that is this here, this inner membrane. That's where the last step, the electron transport chain, is going to happen. And this actually does come up later, and there's a labeled mitochondria coming up later as well. So if you didn't get it yesterday or you didn't get it just now, you'll get it the third time around. All right, so, and this is just a visual of the same thing, glycolysis, cytoplasm. The second step is the citric acid cycle here in the middle, and then chemiosmosis or the electron transport chain is going to be the final step. All right, so here's what happens in glycolysis. Glucose basically gets split in half. Glycolysis literally means the splitting of sugar. Glyco for sugar, and you should remember the word lysis from an earlier chapter, which basically means to break. So all that actually happens in glycolysis is that glucose gets split into 2 pyruvates. So if I was to draw glucose as a 6-carbon molecule, at the end of glycolysis, glucose has been broken into 2 3-carbon molecules. That's literally all that happens in glycolysis. Now, you'll notice it says here that it takes 2 ATP to get it started. But it says here that it makes 4. So it makes 4 ATP total, but it costs 2 ATP to get it started. So the net, and usually on a test they're going to ask you how many net ATP are made by glycolysis, and your answer is going to be 2. Only 2 net ATP are made because it actually costs 2 ATP to kickstart glycolysis. The other thing that's made at the end of glycolysis are 2 NADH molecules. NAD, if you recall, is a coenzyme. The oxidized form is NAD+, and what happens in glycolysis is that 2 of those pick up hydrogens and become NADHs. So again, it means the splitting of sugar, and all that really happens is that glucose gets broken in half. The name of the 3-carbon molecule is pyruvate. That's what we call the 3-carbon molecules. Glucose was the name of the 6-carbon molecule. And then we make 2 net ATP, 2 NADHs. Let me show you, I have it actually here. This, so it means the splitting of sugar. Here's our end products. Again, remember that this is 2 net ATP. On the left, this is actually what glycolysis looks like. So even though all that happens overall is that 1 glucose breaks in half, it's actually a multi-step process. And every step requires an enzyme. So we start with glucose here at the top. Glucose gets broken into glucose 6-phosphate, which gets broken into fructose 6-phosphate, then fructose 1,6-biphosphate, yada, yada, yada. It's a whole long thing. And every step requires an enzyme. If you take like cell biology or maybe biochemistry in college, you would probably have to memorize some of the steps and know some of the enzymes and that kind of thing. For our class, you don't have to know any of that. All you have to know is the overall reaction, which I have here summarized on the right. But I did want you to be aware that it is actually a bunch of small steps, not one big one. And you can also see here, here's where the 2 ATP are required to get it started. So the 2 ATP go in, but then if you look down here, 4 ATP end up coming out. And I'm sure I could also find here, here's where the NADHs are made. 2 NADHs are made down here. So again, that's what it actually looks like, and this is your summary. Now, here's the thing. So this NADH thing is important. NAD, the oxidized form of this coenzyme, is necessary for glycolysis to run. You need it here and here. So that means that if you were to run out of NAD, the oxidized form, glycolysis would shut down. The reason I bring that up is because at the end of glycolysis, the NADHs that were made, they have to get rid of their hydrogens and go back to being NAD+, back to the oxidized form. If oxygen is present, that's not going to be an issue because cell respiration will continue. Aerobic respiration will continue. Those hydrogens will eventually get picked up by water, or by oxygen and form water at the end. If there is no oxygen present, though, this is the reason why fermentation happens. So fermentation, anaerobic respiration, occurs, the purpose of it is to recycle NADH when there's no oxygen present. Fermentation itself, that step after glycolysis, does not make additional ATP. What it does do is it gets rid of the hydrogens by transferring them to something else, not oxygen, and they get recycled. The products of fermentation in bacteria, a lot of bacteria, it's ethanol, which is alcohol. So this is how when they make beer and they make wine, they're basically making ethanol by fermentation. When we go through fermentation in our muscle cells, like when you're running and your muscles start to burn, that's because you make lactic acid. We don't make ethanol as our product. We make lactic acid as our product of fermentation. This is not normally happening. Right now, you shouldn't have fermentation happening in your cells because your cells have plenty of oxygen. But if you're running or you're lifting weights or you're holding your breath and your cells start to run out of oxygen, cell respiration won't completely shut down. What'll happen is fermentation. Your cells will still keep doing glycolysis, and they'll recycle the NADHs at the end of glycolysis by fermentation. Get the NADHs back to their oxidized form, and then glycolysis can keep running. It doesn't make enough ATP to keep you alive for a long period of time, but it does make enough ATP to give you bursts of energy when you're running or lifting weights or you're in that kind of a situation to keep you going a little longer before your muscles would give out. So that's what fermentation is, and that's the purpose of it. The technical purpose of it is to recycle the NADHs back to their oxidized form so that glycolysis can keep going. But again, it won't work for a long time. It actually won't even work in bacteria for a long time. If you were to look at the alcohol content of beer and wine, I don't think you can get any kind of like wine that has a higher alcohol content than like 18%. And the reason why is because the bacteria are doing fermentation and the alcohol just builds up. There's nowhere for it to go and it kills them. So the bacteria themselves are killed by their waste product. And that's why beer and wine are not stronger than they are. Hard liquor is made in a different way. It's made by distillation. It's not made by a living organism. The other place where you'll see fermentation happen is if you ever make fresh bread with yeast, because the other product of fermentation in bacteria is carbon dioxide, and that's what makes bread rise. So if you've ever made fresh bread where you cover it with a damp towel, and you let it sit for a couple of hours, that's the yeast going through fermentation. Technically, it's also making ethanol. It doesn't make enough that you could like eat bread dough and get drunk or anything, but it does make ethanol. And then when you bake it in the oven, the ethanol burns away, but all the little holes are coming from the fermentation that happened while you were letting it sit. So let me go to this next slide here. This is just showing a really simple overview. So glycolysis happens whether oxygen is present or not, whether you're a bacteria or whether you're a person or a plant. At the end of glycolysis, the pyruvate either will go through fermentation if there's no oxygen present. That'll recycle the NADHs, and you'll make either ethanol or lactate. Lactate is another word for lactic acid. If oxygen is present, we're going to go the other direction towards the citric acid cycle, which is the other part we're going to talk about today. Two products of alcohol fermentation, not just ethanol, but also carbon dioxide, and again, that's what makes bread rise. This is actually what it looks like. You do not have to know this picture. The reason why we don't make alcohol, because that would probably make exercise way more popular if we made alcohol instead of lactic acid. You just get drunk when you worked out. Is because we have a different enzyme. We have lactate dehydrogenase, so our pyruvate turns into lactic acid. The bacteria have these two other enzymes, pyruvate decarboxylase and alcohol dehydrogenase, and that's why they make alcohol. And actually not all bacteria make alcohol, because like when milk goes sour, those bacteria are basically souring the milk. They're making I think they're actually making lactic acid as well. And there are some bacteria that make acetic acid. Vinegar is also made by some bacteria. So it's not always these two products, but these are the two that they usually want you to know. That is ethanol in many bacteria, and then lactic acid in animals. Not just us, but all animals. Okay. Again, why is this beneficial? Because it does provide you with quick bursts of energy, but it does make it burn, and you will become fatigued. You can't go very long running fermentation, but it does keep you going. You know, gives you that burst of energy, so you don't collapse right away if there's no oxygen. If oxygen is present, the reaction is going to continue down the aerobic pathway. The oxygen will pick up hydrogen and become water. You will harvest about 40% of the energy from glucose. The remainder of the energy from glucose will actually be lost as heat, which doesn't seem very efficient, but they say that it's actually much more efficient than most man-made processes. That even, for example, running your car, I think you only capture about 25% of the energy from gasoline, and the rest of it is lost as heat. So it's actually much more efficient than, gasoline running your car. We're actually extracting more energy from glucose than most man-made processes are able to do. The rest of the steps that are going to happen, the pyruvate is going to now cross in the mitochondria. Everything else that's going to happen in cell respiration is going to happen in the mitochondria. When it goes into the mitochondria, the first thing that will happen is the citric acid cycle, also called the Krebs cycle. We'll talk about that one. That's the last one we'll talk about today. And then the final step is going to be the electron transport chain. And that is the last step, and we'll talk about that one tomorrow. So mitochondria, again, everything else is going to happen in the mitochondria. It looks like it's got an outer membrane, and then the inner membrane is like highly folded. It's all squiggly. The inner membrane is also called the cristae. So you see this right here, cristae. That is this inner membrane of the mitochondria. The matrix is the center. That's where the Krebs cycle is going to happen. And then the intermembrane space is this gap here. In between the outer membrane and the inner membrane, all of this is called the intermembrane space. And we'll actually come back and talk more about the intermembrane space and the inner membrane tomorrow. Today, we're talking about the Krebs cycle, and it happens in the matrix, which is the middle part. All right, and I can put this slide back at the end, so if you didn't get the structure of the mitochondria, I can come back to it. I just want to go through the citric acid cycle really quick. So the citric acid cycle is also called the Krebs cycle. At this point, when the pyruvate goes into the mitochondria, by the end of the Krebs cycle, all the glucose has been broken down into carbon dioxide. And all the hydrogens from the glucose have been sent to NAD and FAD. So by the end of the Krebs cycle, all your glucose is gone. All your glucose has basically become carbon dioxide, and the hydrogens from the glucose have all gone to NADH and FADH2. The electron transport chain doesn't actually break down glucose. By the time things get to the electron transport chain, the glucose is completely broken down. All the electron transport chain is going to do is work with these hydrogens. The carbon dioxide is a waste product. You're going to basically exhale it. So wrapping this up. So again, at the end of this, at this point, all the sugar has been completely broken down. The carbon and oxygen have become CO2. The hydrogens are all on the coenzymes. Here's what it looks like. Again, just like glycolysis, actually this is just showing the step where it enters the mitochondria. This is what it looks like. Again, just like glycolysis, it's actually a whole bunch of little steps. And the name citric acid cycle comes from the fact that the very first thing made is citrate or citric acid. So that's why it's called the citric acid cycle. And it's called the Krebs cycle after the person that discovered it. So this is what it looks like. It's actually 8 steps long. And one last thing, it does also make 2 ATP. And I forgot to add that, so please make sure you add that in the notes, that the Krebs cycle makes 2 more ATP. So at this point, how many net ATP has cell respiration made by the end? Only 4, because it made 2 net in glycolysis, and it makes 2 in the Krebs cycle. It's only made 4 ATP. In the end, we're going to make about 36 to 38. So all the ATP, almost all of them, are going to be made in the electron transport chain. Very few have been made up to this point. Yes? No.